Okay, hello, everybody. Welcome to another exciting session. We have our own Dr. Hong Han here to talk about some exciting new research that has just landed at McMaster. I'll just give a brief introduction on behalf of Sheila, who unfortunately couldn't make it today. So she received her Bachelor of Science honors degree at the University of British Columbia and her PhD degree in the Department of Molecular Genetics at the University of Toronto, under the supervision of Dr. Benjamin Blencow and Dr. Jason Moffat, who I think will be coming here probably in the near future to give a seminar. During this period, she received over 20 awards and honors. Immediately after her PhD in 2016, she was honored as the first recipient of the prestigious Donnelly Home Research Fellow Fund at the University of Toronto. This allowed her to become a semi-independent investigator to develop projects and supervise a research team. In addition, her research has been supported by funding from CDCR, Simons Foundation, and MyTax. Dr. Han is currently an assistant professor at the Department of Biochemistry and Biomedical Sciences here at McMaster, and a principal investigator at our beloved Center for Discovery and Cancer Research at McMaster. Her lab works at the interface of cancer biology, RNA, and multi-layer gene regulation, as well as high throughput technologies. Dr. Han and her team have pioneered developing and applying several integrated technological platforms for large-scale genetic drug screening and ultra-high throughput single-cell profiling. Leveraging the power of these systematic experimental and computational approaches, together with in vitro, in vivo, and patient cohort studies, they discover multi-layer regulatory mechanisms underlying cancer progression and RNA multimodal therapeutics for treatment-resistant cancer. Her team strives to create a collaborative, supportive, and stimulating environment for multidisciplinary cancer research, and I've considered applying to her group myself. So without further ado, Professor Han.
Thank you so much, Anthony, for the introduction.
No. Right. And then put up in.
That's all we need to get started. So yeah, also thanks everyone for coming today, and both in the room and also online. And I'm very excited to discuss some of our recent work, especially the multi-layer sulfate control in both development and cancer. So as many of us know, cancer is the leading cause of death in Canada, and 2 in 5 Canadians are expected to develop cancer in their lifetime, and 1 in 4 Canadians is estimated to die from cancer. So these numbers are devastating. However, because of the great effort from our large team, including clinicians, scientists, and many other parties, we did make a change and can bring hope. So for example, since 1970, the cancer survival rate have doubled. And with the emerging current technology and rapidly evolving cancer research and management, it's actually important to see what we can do in the next 5 years, 10 years, or 50 years as a team. So cancer has been long recognized as a caricature of novel tissue renewal. So it can occur in any part of the body and involves complex interplays with the surrounding tissues. In order to fully understand the mechanisms that drive or contribute the progressions of cancer, we need to look at in a systematic manner. And also, we can learn from development, especially to study the dynamic process of treatment-resistant cancer at different disease states. So both development and the cancer are actually very complex process, but we can organize these into 3 axes. For example, the coordinate multilayer gene regulations can play critical roles to control cell fates and the temporally and also spatially. And this can actually regulating the key cellular transitions in both development and the cancer. However, a main challenge for study this complex system is most time of our current works, we study 1 gene layer at a time. And it will be great. We can link in these different layers and also track these change over different time and the space. So by taking advantage of this complexity, we can actually better learn the genotype to phenotype relationships and also identify the most robust driver or regulator event. So as a system biologist, I'll discuss what's the approach we can use to study these important questions in a timely and a systematic manner. So for today's talk, I'll just give a very brief introduction, and then I'll discuss 2 recent of our projects for neurogenesis and also glioblastoma. And these will further leading to the current and also future research in my team here at McMaster. So brain is the most complex organ in our bodies, and during the brain development, there are multiple waves of gene regulations, and that is required to control the key cell phase transition. For example, from the neuroepithelial cells to progenitors, and also different subtypes of neuron and glial cells. The size of the brain is also significantly increased during the development and involving complex cellular organizations and interactions. So on the other hand, tumor is a dysregulated developmental processes, and it represents a highly complex and rapidly evolving ecosystem. And I want you to remember that message. I'll go back to this and at the end of the talk. So we know the tumor actually display extensive heterogeneity, and also both the tumor and the microenvironment involves rapid evolutions during the progress of cancer, like such how they respond to the treatment, and in certain cases, leading to recurrence and metastasis. So it's actually important to study the diverse populations in both the tumor and the non-malignant compartment, the microenvironment. And as I mentioned, in addition to the individual layers, I also interested how these different layers coordinate together to control important biological processes. So just using 1 example here. So using RNA sequencing data from a diverse cell types and the tissues, and I identified the MBO RNA binding proteins as a master regulators for a large network of embryonic stem cell alternative splicing events. So these events regulate different pathways, including key chromatin regulators, transcription factors, such as the very famous stem cell transcription factor, ARC4, NANOC. So together, this provide one of the first evidence splicing factor can coordinate multi-layer gene regulatory networks and play a central role in biological process such as pluripotency and the reprogramming. So interestingly, the MBL regulate alternative splicing networks also play an important role for tumor stemness and the progressions. So low level of MBL expression has been implicated in multiple cancer types, including glioblastoma, gastric, and also breast cancer. And when MBL level is low, it's actually associated with like poor overall survival and also increase the chance for relapse and the metastasis. So more broadly, actually alternative splicing can impact every hallmark of cancers, such as proliferation, apoptosis, immune response, and also angiogenesis. So I think even though there's a complexity for different layers, but it's really, this is one example. We can actually take advantage of this complexity and using that as opportunity to learn new biology and develop therapeutics. So what I mean by that, so as I mentioned, alternative splicing regulation represent a key layers. And I promise I won't get into the details for that. But when you look at alternative splicing as an overview, you probably already noticed some of your favorite components, such as RNA sequence or structure, signaling pathways, chromatin transcription, and the non-coding RNA regulators. So that means even though there's a complexity associated with alternative splicing, but there's also means there's many ways we can use it to modulate not only isoforms, but also using splicing to monitor expression levels and the protein expressions. So the key take-home message from here is like more than 95% of human genes are alternative spliced. And when we look at other human disease, these mutations, 15 to 50% of these disease-causing mutations affect alternative splicing. So that means when you study your genes and in a developmental or disease context, it's worth to look at its isoforms as well. So here, I just want to give you a very quick overview. And so there are three key interconnected components for my research program. So typically we starting with pressing questions in development, cancer, or other disease. So these biomedical questions really drive us to developing new technologies, especially when the current technology have limitations to study these questions in a timely or systematic manner. And in parallel, we also developed bioinformatic pipelines to further increase the efficiency of biological discoveries. And importantly, these high throughput studies is really help us to generating hypotheses and leading to important biological discovery and also potential therapeutic targets. So actually, in addition to high throughput study, we also go deep and to study the function and the mechanisms of the target or isoforms we identified. And this is going to be together leading to this power cycle of discovery in my team. So for the next part, I'll using a neurogenesis project as an example to see how we can using a functional genomic screens and also ultra high throughput single cell profiling to identify a large networks of regulators that plays important role to control neurogenesis. So as already mentioned, brain is one of the most complex organs. So when we look at in different brain regions, and there are specific events such as alternative splicing and expression is associated. And when we look at across the time of a neurodevelopment, there's also different dynamic patterns of important regulation events, such as decrease or increase or other dynamic patterns during this multi-stage neurodevelopment or neurogenesis. So to study these events individually, it requires a lot of effort. And it's actually, when we do that, it could be also potential missing the big picture or coordinations. So in order to better understand the multi-layer regulatory mechanisms underlying a complex mechanism such as brain development, I developed this highly multiplex and a quantitative like a screening approach called splicing. So it represents systematic parallel analysis of endogenous RNA regulation coupled to barcode sequencing. So in brief, is actually using optimized multiplex RT-PCR reactions, we can simultaneously amplify up to 100 to 200 or like RE regulation event interested. This can be expression event, splicing, and also in a current screen performing in the lab and the collaboration with other teams. We're also including circular R events. So there is many creative ways we can design these panels of sparsing events. So following that, we actually design a dual barcode system. We can monitor each wells or each treatment. So at the time we initially developed sparsing, Illumina, everyone knows, is the popular option for sequencing. They offer 96 barcodes. And we know we need more combinations. So we actually design our versions of own barcodes, initially with 768 options. So that's actually flexible barcode. It's very important. So since the initial development, we've been using this platform to study different developmental biology, but also neurological disease, rare disease, and also during the COVID time, we're able to very quickly change our platforms in a couple of weeks and using the barcode to detecting COVID, and is actually running thousands of reactions every day at Mount Sinai Hospital in collaboration. So I think this platform is really offer a powerful approach, and we can use to study different biological process in both novel and the disease context. And also the resources we're generating here. One thing I think there's one version of Sparsic, we're generating the RU is enough to performing 30 or 50 screens. So that is really like a lot of potential we can using it. So go back to the question. Now with the Sparsic screening platforms, we can really study all the spatial temporal neural regulation events together. So in that particular screen, we actually monitor close to 300 splicing and expression events. So we perform this screen in a neural progenitors. So that's give us the chance to study both early and the late regulation of neural differentiation. And also the perturbations we use in this screen is actually ICNR knockdowns of splicing factors, chromatin transcription factor, and a short list of signaling pathways. But there's really no limitation for perturbations. You can couple with a CRISPR knockout and inhibition activation, but also can couple with a drug treatment. I'll also further commenting that later at the top. But with this highly multiplex and the quantitative screens, and we can really generating an in-depth readout. And from that, just from a single screen, we can identify hundreds of positive and negative regulators that are important for neurogenesis at different stage. And this study was recently published at Molecular Cell. It's really representing amazing teamwork and also bring many of my long-term collaborators and mentors together. So, but we don't stop there. We actually go beyond. So the advantage of Sparsic is we can actually combine different screen we perform, such as the current neural screen and also a previous stem cell screen. So with the Sparsic team, especially Nick, Andy, and Eric, we also integrate the screen data with both bulk and the single cell RNA sequencing data. So we can generating these multi-model integration networks, which can tell us better about the specificity and the mechanisms of all the factors we identify from a Sparsic screen. So for example, in one cluster, we identified a negative regulator for neural splicing, which we call RBM38. And in another cluster, we identify a positive regulators for neurogenesis and the neural splicing. So when we're looking at the single cell data we use for the integration, and here actually is showing a UMAP presentations of a single cell data at different stage of in vivo mouse development, and each dot representing a cell. But maybe just focus on the neural cell populations for now. And you can see for the RBM38 is lowly expressed in neurons, consistent with as we identify as a negative regulator. However, for PUF60, and it's expressed everywhere in different cell type and tissues, and we wonder how it can give the neural specificity. So we actually look into deeper into PUF60, there is exon 5 adjacent to the first RNA binding domains. And this exon 5 is highly conserved during vertebrate evolution. And importantly, it's neuroregulated. So that's the way how we're thinking is regulated. And then we perform a series of experiments to further understand its mechanism and the functions, but make a long story short. So one of the experiments we're performing is using neuroprogenitor cell lines expressing the POP60 isoforms, and we perform AP mass spec to locating the global protein interaction. So from the AP mass spec, we found the POP60 minus exon 5 isoforms actually preferentially can enhance the binding to splicing cofactors. So further morphology and also bulk RNA sequencing analysis further reveal the POP60 isoform have a stage-specific role during neurogenesis. So for example, for the minus exon 5, it's actually important for early differentiation from embryonic stem cell to progenitors, while the long isoform is required to the later stage from progenitor to immature and mature neurons. So this is nice, but also leading to the question to see, is POP60 having cell type specific roles? And so that way we need to analyze different genotypes we're generating and also across different time points, and the commercial platforms become limited. So that's actually we're thinking alternative ways. So many of you here probably hear about single cell, and even some of you already using commercial platforms as 10-time genomics. But the high cost associated with commercial platforms, it becomes a main barrier for many of us as academic research and also for clinical researchers. So what is really happening in that machine is forming the droplets containing the single cell and barcode bead. But that's not the only way we can barcode the transcript from the single cell or nuclei. So actually the concept is very cool to use. It's called single cell combinatory indexing. That's also the method we use to building our single cell platform. So the key for this method is the 3 levels of indexing. And for example, in the RT-PCRs, you can add in the barcode, each one's getting different unique identifier. And then we putting all the cells, redistribute to the second levels, and then they get a different barcodes. And then this pull and the split process is performed again at the third level. So you can imagine this is a lottery system. So at the 3 levels, each level we're using 384 barcodes. So this gives us a huge combinations of a 3 barcode. When only using a small portion to label 3, like a couple million cells, and each of the single cells will get a unique combination of 3 barcodes. So to summarize that, this is really the ways we build our in-house ultra-high-throughput single cell platforms. And since the initial development, we've been really increased the throughput. It's give us a cost-effective and approach to performing like a large scale of single cell profiling. So with a team of 4 or 5 people, we can actually profiling up to 100 samples in a single experiment. So I think really behind these numbers is we can using this approach to answer the biological questions we're interested in, such as development or cancer, in a very different way now. So at the same time, we're also developing our bioinformatic pipelines, and I won't get into detail for that, but one of the paper we just published the first few modules, but we have many other modules that can performing a typical single cell analysis, but also in-depth analysis, such as the integration I showed earlier. And my team here will be continuing building these analysis modules and sharing with the community. So now go back to the questions. Here is just a quick summary for different genotypes that we want to analyze. So we can delete the exon 5 in POP60, but also using a homogeneous directed repair to insert a CD inversion. So the exon 5 is always included. So we can add it 1 copy or 2 copies to generating different homozygous or high clones. So here it's really just a quick overview of single cell data after integrating all the genotypes and the time point we profiled. So importantly, from these high resolution of single cell data, we can learn the differentiation trajectory, but also the cellular heterogeneities associated with this in vitro differentiation. For example, at neuron differentiation day 7, we actually identified different subpopulation of neurons, but also glial cells and oligodendrocytes, and some very rare populations of non-neural lineage cells. So this is actually very important for us. So in addition to building the cellular maps, we can also generating a transcriptomic gene networks. So in this version, each dot represents a gene versus a single cell. So using this approach, we can actually look at how these genes are co-regulated and enriched in specific cell types and or different subtypes as well. I'm only going to using 2 examples to show how it can tell the mechanism and the biology. So 2 gene pathways are highlighted here. Is epithelial to mesenchymal transition and also extracellular matrix pathways. So these 2 pathways highlighting in the gene networks is very specifically expressed in the vascular cell population or the fibroblast-like populations. And we can further link these 2 different genotypes. For example, for the plus exon 5 is actually promoting neurative pathways, while for the minus exon 5 it actually increase the EMT and the ECM pathways. And we can also confirm these observations by looking at cellular abundance difference. So for the plus exon 5, which is play important role at the late stage of neural differentiation, is promote neural lineage differentiation, but inhibit these non-neural fibroblast cells, while minus exon 5 actually is promoting these rare non-neural cell populations. If you remember the Hess cone I mentioned earlier, so it's actually showing intermediate effect, so showing the robustness of this data. And so PUF60 is one of the examples we identified from the SPAR6 screen, and many of the factors we identified not only play a role in neurodevelopment, but also associated with certain disease, such as this case for PUF60 is Verheyen syndrome. And also PUF60 had been implicated to play an important role for cancer progression. So that's also further highlight the say when we study a gene function, maybe we should also consider whether there's different isoforms associated that genes. So just quickly summarize these parts. So I've shown we can using the highly multiplex functional genomic screen coupled with a single cell approach to identify a large network of regulators that are important to different stages of neurogenesis. And also we follow up very deep function and mechanism study to 2 of the factors. But this is really just a limited factor we can follow up from 1 study. So from the screen, we're actually having a network of a few hundreds factors that is interesting to study in the context of development or disease. So that will be forming the basis of our future research in the lab. So for the second part, I'll switch gear, but still in the neural context to see some of the works we've been done to understand the mechanisms underlying glioblastoma, especially at the recurrent stage. So glioblastoma, I don't have to introduce more here. It's the most common and lethal primary adult brain tumors. So even with the maximum standards of therapy, the patients still relapse between 7 to 9 months. And I guess the characteristic for GBM involves is having their extensive molecular and cellular heterogeneity, and also occur in the brain is really having a very immunosuppressive microenvironment. And also very important is to see, even though in like a previous study mostly focus on 1 stage, but in order to really understand the recurrence mechanism, we need to look at different stage of this disease before and after treatment, and also beyond what is really causing the difference at the recurrent stage. So very interestingly, glioblastoma actually use and hijack many of the neurodevelopmental pathways as a way to control their tumor cell states and the plasticity. So you can see there's different subtypes of GBM tumor cells. And then actually collaboration with across Canada teams, especially Dr. Sheila Shin's labs, Jason Mark's lab, and Gallery today's labs. We're actually analyzing patient-matched pairs of GBM, but also look at additional GBM mouse models or xenograft models. Together, this give us a better picture for biomarkers and also the key regulator, what's their function and mechanism to regulating GBM in order to generating the next generations of like therapeutic targets. So just using a pilot study as example. So here we're actually profiling 18 patient matched samples. As I mentioned, the single cell platform we're using is giving the capacity. We can look at all the cells in the GBM ecosystem. We don't need to do any cell sorting before the single cell profiling. But that also means after profiling, we need to distinguish what are the tumor cells, what are the non-tumor cells. So we're actually developing a multi-level scoring system to help efficiently and robustly identify these tumor and non-tumor populations. So when we look at the tumor cell populations, the first thing we do is gene differentiation, differential analysis. We can actually found gene signatures that is distinguish primary and the recurrent GBM. So that's a good indications of the profiling is very robust and we can find interesting things associated at each stage. And also we look at pathway levels and to see what is associated as a primary and a recurrent stage. Not only using the initial profiling cohort, we can further validate these discoveries using independent validation cohorts. So one observation we found is the recurrent tumor is more proliferative and the immunological colder. So it's probably consistent with our expectations. But when we look at the non-tumor cells, including many diverse types of cells from the microenvironment, and we can actually identify many subpopulations. Very interestingly, we found from the microenvironment cell populations, they play important role at the recurrent stage. So there are cell populations that have decreased or increased. So I think this is really giving the opportunities. Now we don't have to only look at the tumor because it's really function as the ecosystem. We can look at all the subpopulations from the tumor and from the microenvironment and also their interactions to better understand the GBM biology. So I just want to using another mouse model for GBM to highlight to see how we can study GBM or other cancer types at ecosystem levels. So here actually we can analyzing a whole brain regions using the single cell platforms we built. So we can analyzing the initial site of injection, but also secondary sites when the tumor is migrate. So from this UMAP, you can see is actually from a single experiment. Actually, there's more samples analyzed in that specific experiment, but just this one, we can look at all the brain tumor cell, brain cells, tumor cells, and the immune cells together. That is give us a more comprehensive cellular interaction maps. So as I mentioned, using this approach, we can really analyze different GBM models, such as more mesenchymal versus developmental-like GBM models, but also look at their dynamics from in vitro to in vivo, and also look at the region difference. And also using machine learning approach, we further integrate the patient data with the mouse models or other models, and that will help us to identify consensus key regulators and driver events that will further guide us downstream function and mechanism studies. So that's how next, subsequently, we're generating the knockout cell lines and inject to the mouse brain and to further study the tumor and also its impact to the microenvironment interactions. Not surprisingly, among these hits, including RNA binding proteins. So for one of the RNA binding proteins, we actually, when we knock down these factors, we see a significant benefit of survival. So that's a good indication of hit we follow up. So as a quick summary for this part, so we're really developing a ultra high throughput single cell approach, which allow us to understand the GBM ecosystem evolution, but can also apply to study many other treatment-resistant cancers. So using this approach by analyzing different time points, different location of the tumor, different models, it helps us to identify key biomarkers and also regulators, and which can be further studied for its function and also developed to as the new potential therapeutic targets. So I think for the two projects I discussed, I hope I give you an idea to see what kind of biological questions we can answer and also what approach we can develop to more efficient study this one. But I think this is just a quick summary of my research journey or background. So in addition to the neural lineage, we also actually study mesoderm lineage, especially for the blood progenitor and the immune cells differentiation. You probably can know why that's interesting. That could be tell us better when we're thinking about immunotherapy or needing certain cells to treating cancer. So in addition to GBM, my team is also studying prostate cancer and small cell lung cancer, which also aggressive type of cancer they developing treatment resistance. So this is really leading to the research in my lab at CDCR right now. So for the last part, I'll just highlighting the current research theme, but also broader applications and the collaborations. So as Anthony mentioned during the discussion, and my lab is really working at the interface of cancer biology, RNA, and multilayer gene regulation. And also we develop high throughput technologies, both experimental platforms and the bioinformatic pipelines. So I think, so I already shown you the single cell profiling. We're starting from the transcription levels, especially gene expression. But right now we are further expanding this to co-profiling of splicing, expression, and the mutation at RNA levels. But also we're going to build, like adding additional modules to the single cell pipeline to analyzing epigenetics, such as using single cell ATAC-seq at performing at the single cell levels. So as I mentioned, the single cell combinatory indexing methods is really like versatile in the way you can be really creative. The 3 levels of a reaction, and you can be applied to any omics you can thinking of, but also it's compatible for spatial transcriptomics. And you can not only look at all the cell population, but know exactly how their physical locations and interactions. Actually, just 2 weeks ago, not only we can doing single cell transcriptomics, we can even do single cell ATAC-seq is the chromatin for with the single cell, but also spatial resolution. And then I already shown you for the splicing application for genetic swings, but it can be also applied for a large scale small molecule or other type of a drug reagent screens. And at the same time, it's actually interesting if we can integrate these molecular profiling and the screening data with medical information. So this way together, we can build in a digital biobank in addition to the physical biobank. And this way, all the information can be used by different researchers and both basic scientists and the clinicians to using this multi-model information to better selecting biomarkers and also potential therapeutic targets. So actually one of the research theme, actually almost every of my team members right now is looking, we started with the bioinformatics. We're interested in alternative splicing regulatory networks in different cancer types. So we can actually look at in pan cancer, which means almost every cancer types we can look at in. In addition to the splicing event networks, we can also look at its main regulators, such as RNA binding proteins. So when initially we do the splicing studies, we having about 1,500 RNA binding proteins, but recently the team have expanded to 6,000, but very likely we're going to using the top 3,000 for additional splicing screen to understand local versus metastasis prostate cancer. And this, the other thing is, as I mentioned, is a continuous study for glioblastoma. So in addition to just for the primary and the recurrence, and with the nice collaboration here, and we can actually go beyond at the end stage to look at the spatial heterogeneity as well. And also for the different layers, we can start from the expression, but really adding additional layer of information by co-profiling or integrations. So I think here as a teams across different labs, we can really to better understand to using this technology to study patient tumor or further study their mechanisms. And also for the last part, as I mentioned, the splicing can be using for different types of genetics and the small molecule screens. So in addition to the splicing project I already mentioned, my team before starting from Toronto and now at McMaster, and we've been really generating different panels of events we are interested in. It's starting with neurological and muscular disease, but now we are more focused on different type of a cancer event, such as gene expression, alternative splicing, but also like other circular RNA event or any other event we can monitor. And so in that way, you can imagine it's a kind of a common challenge, not only for academic lab, but also for pharmaceutical companies. When we only look at one disease target, and even we can screening like up to millions of small molecule, but the success rate is actually low because it's a very high risk. We don't know what we're going to discover. But using splicing, if we can analyzing 100 or 200, the most interesting and the related disease events, is significantly increase the success rate. But also by analyzing different events, we can better understand the specificity of the compounds we identify. And even just from the initial primary screen, we can learn the potential mechanisms. We can also integrate the small molecule screens with the genetic screens we perform. And it's right away telling the potential target or mechanism. So I think I'll go beyond that current project we're doing in the lab. I think this is kind of the visions maybe 5 years or 10 years from now. I think it's really the great opportunity at CDCR and also rather at McMaster and also go beyond, working together with different expertise, and we can applying these cutting edge technology to study patient sample or different cancer models, and also the same times performing genetic screens and real-time therapeutic discovery screens. And I think in the near future, maybe we can also couple that together with clinical trials, because in many scenarios, and there is a negative result from clinical trial. But using this approach, we can learn even it's a failure, like what is happened, how we can do it better in long term. So I think that's really just a lot of potentials. And I will also, will be nice, we open the discussion to see maybe what type of samples you are interested in and how we can work together. And I just want to bring this back to see at the beginning of the talk, I highlight cancer is a highly dynamic and rapidly evolving ecosystem. So to study and to treat, prevent, or conquer cancer, maybe we can also building a highly collaborative and rapidly evolving research teams. So actually my journey, as you just see, starting from system biology and developmental biology, more recently, I was more involved in cancer research. So I really thanks all my collaborators and colleagues, which is working in the cancer field from different angles. It's from them, I learned so much about different angles of cancer biology, like different models and immunology, like T cells, NK cells, and also like the drug discovery, and also from the clinical part. There's many information actually we can building link from the clinical such as MRI or like other pathology slide. And then we can link to the molecular information we gain from a basic research lab. And so even go beyond that. So I also learned a lot and actually enjoy that process to actually collaborating and interacting with researcher and clinicians that are not, they work in the traditional way, not a direct related cancer field, such as organ transplant, burn injury, or infectious disease, and from bioinformatics to artificial intelligence. Even though in many scenarios, we actually study a different political questions, but actually we actually having the similar concept or methodologies. For example, we probably all sharing the like system levels of complexity, and there is a very rapid evolutions involving many of these complex process. And in many scenarios, we have to look at that disease or development as ecosystem level. So I think that just as a quick view to see, maybe it's definitely is only highlighting a few like regions, and from this room and from the collaboration here, it can really like bring this research ecosystem, and you can add in new field here, and maybe we can learn from engineering and stuff. Acknowledgement, just a huge thanks to all my like mentors, collaborators, and also the two teams, particularly working on the splicing project and also the single project for glioblastoma. So this is the current teams in my lab. They are really here to changing from an empty lab to a dynamic team. So some of them have known me for many years, and some of them they joined in the labs for 3 months or 3 weeks, but they have been making a huge difference and making both like a experimental experiment and all the experiment part and bioinformatic analysis. And so that's really like make me excited here. And also thanks to every of you here, because I feel so much welcome and the support from CDCR and from my department, from the faculty and university level. I think it's every of you making I fully enjoy the past 5 months here, and I'm looking forward to the next stage.
Thank you. That was a great talk.
That was sort of force, so thank you. I'm going to begin with a simple question, which is, how would you use science seek for spatial profile? Because spatial profiling is usually probe-based.
So actually, how it works is actually, there's already a team from Seattle is creating a slide with oligos and on the, so that's actually the way we can couple, or it's compatible with the sky method to getting the spatial information. So you are right, it had to be slide and having imaging components involved. But actually the sky is actually help to getting the spatial information with the cellular, because other spatial transcriptomic approach, they can limit it by the resolution, like certain micron regions, but they cannot tell the cellular information. So I think that's actually something interesting. And also we can make the probe in a way to see if we're spending to splicing junction, we can also learn like splicing isoform information. But
What's your rate?
yeah, so it's on a slide.
I guess a harder question is, when you have one target, it's hard to find any kind of drug. When you've got 10 targets, it's probably 1,000 targets. When you've got 150 targets, it's probably 1 million targets. So it's great that you suggested that to simplify drug development. I would actually argue. Are you considering any type of pipeline that would allow you to match up the nodes that you discover against databases of drugs already in order to try and facilitate the process and say, you know, what is the most likely to go after because it's druggable already? There's so many, there are many targets that you know full well are undruggable, or at least currently undruggable.
Yeah. So I really like to,
Sorry, I just wanted to say, Han, for the people that are virtual.
Yeah.
If we could attempt to repeat the
Well,
questions.
it's fine.
to some
Mine
extent.
carries across.
Can the people, so the people that are on Zoom, you're gonna have to ask questions out loud, just the way we have it set up. So let us know if you can't hear what the question is also on your end. Sorry to interrupt.
all right, that's good. I forget to repeat. Also,
No, I
I'll
didn't.
launch this slide. So actually really like to have that discussion to see a short answer is we try everything to be efficient and also more robust to finding a therapeutic target. But I think let me starting with one direction and then I'll go back to the regular direction your question asks. So the initial challenge one we have when we do the high throughput profiling study, it doesn't matter it's a bulk or single cell. And we actually encounter this dilemma to say after a profiling study, we found hundreds or thousands potential biomarkers and regulators. And we ask our the question, which are the most important? And in the traditional way, we try so hard to finding the top one or top two and thinking that's the best one and making all our efforts. But then in many scenarios, it's not true. And we actually regret we didn't select the other one. So that's the multiplex screening platform in that angle helps to see, it gives us a chance and we can look at in the top 100 regulators. And it's different from profiling or even like genome-wide association study. This gives you the information is the potential implications or association while when we really knock down a gene, and then we can actually really know what is the change after perturbation. That helps us in a more systematic way to testing all these factors. So then that helps us to further narrow down one or two. But then I guess the drug screen is a different scenario to see, like if you have the top number one hit to see there's a disease only causing by that gene. Actually, there's a SMN1 is a muscular dystrophy disease. It's only causing by one gene. So you probably don't need many of the other target for that disease. But the platform we're trying to use to see maybe in that scenario, we can study more than one disease together, like in the muscle context. It's not perfect for any like in vitro system or even in vivos to see if it's a neural context or muscular. We have to study neural-related disease or muscular disease. But the idea to see we can maybe study top one or two targets from each disease, but 100 disease together. And that's the way to see we look at the potential chance. And I guess another way I think we're also trying to use, because currently there's many databases we can use. There's, for example, our 1000 small molecule profiling database. So we already know from that database to many of the compounds commonly used, what is the transcriptome change after. Actually, for that database, they focus on the what around 1000 genes is most interesting. So we're trying to learn from that information as much as we can to see which small molecule library we should be screening is going to be more successful, and what type of event we should monitor, but also considering like a specificity control event. So I think that's all together is important. And I'm sure there's many more things from the drug discovery fields we can learn from to doing it better.
So you're pretty much saying, Hung, that in this type of approach, you've got a better chance of getting maybe a more relevant target because you can screen so many different pathways at the same time. That's pretty cool. Any other questions from the audience? Or from the cloud? I have a basic question. On one of your earlier slides, I'll fill some space. We'll submit if somebody wants to conjure up some questions. Why are there 3 primers added per well? I didn't quite follow the process there. I think it's one of your earlier slides. It's
Is
like a basic question.
is the splicing barcode, or is there 3 level of single cell barcodes?
He realizes that you pull the cells up, you distribute them, you give them a barcode, or back up, distribute them again, another barcode. So every time you get another barcode, the odds of having any cell We include the same barcode, it's progressively less.
Okay,
Is that-
I
is that
see.
how it's done? You can pick
Yeah,
them up, put it
so-
back down.
Yeah, so that there's single cell 3 levels barcode, because for each levels, you're getting unique barcode, but all the cells from that walls is very likely go to different walls at the second level. And then you pull all the cells from different wall, redistribute, and they will go to different walls again. So exactly the same cells go to exactly the same walls from level 1, level 2, level 3 is very rare. So that's actually how you're getting the unique combination.
okay, that's really cool.
I think
Oh,
Tobash
Oh,
having a
yes.
comment. Oh, please go ahead.
Oh, hi. It's great talk and great methodology. Fantastic. Can you just comment about again what would be what the maximum number of cells you would be able to analyze in one of those like runs essentially of your methodology or in this workflow? And again, what the depth of genes or how many genes have you been able to analyze then from the sequencing? So essentially, how does that compare to essentially the 10x pipeline?
So maybe I'll repeat the question for the audience. So Dr. Berg is asking to see, using these Sky methods, what's the capacity in a single experiment, how many single cells or nuclei we can profile? And then on the other hand, it's always a detection question for single cell, how many genes we can capture. Is that right? Okay, perfect. So I'll answer the first one. So as I showed, so with the current combinations, we can barcoding up to 2 or 3 million single cell or nuclei in a single experiment without problem. But we have to make sure it's only about the 5% of the total combination is used. The more we use, there's increase the chance of like same barcode from different cells. So the answer is about 2 to 3 million. We want to stay with below 2 million to be safe, but also you can increase the barcode number from each level to increase that. But I think the current bottleneck is with sequencing not single cell, because we're already using the largest sequencing platform, 8 billion Illumina-seq. And then for the gene question, that's actually very important for single cell, because at the early stage or at even current stage, single cell is already being optimized, but still like from each individual cells, depending what's the type of cells and what's the tissue they coming from, you can get in from a few hundreds up to like 5,000, 6,000. That's the typical range. And some of them we know is real biological difference. For example, NK cells actually having very specific set of genes expressed. That's why they have smaller numbers. But in general, there's a detection issues. There's many ways we can actually optimize. And one way actually we're quite interesting, because most of the single cell profiling is focused on 3 UTR. But if we profile the whole transcript across the transcript body, that's another way can increase the capture capacity, but also it's putting the way we need to sequence more and the cost more. So it's always a balance, but definitely single cell field is like changing so much every day. You're probably seeing like 100,000 papers each day about single cell. So it's always keep changing. But right now, it's also because so much data has been published using commercial platform like 10x Genomics, and those reference map is already creating. When you're having a reference map, and you don't have to sequence that deep, because it's help, even you sequence that shallow, you can identify all the like cell types you're interested in.
Great.